GMOs are bad and will eradicate humanity.
Well, it turns out that's not true, and later in this video I'm going to explain why. But what is this CRISPR? It's that method for altering DNA, and it's also a fairly complex concept, so we are going to watch the best video on the internet that explains it. This video is by Bozeman Science, so shout out to him.
If we break apart the acronym, it stands for Clustered Regularly Interspaced Short Palindromic Repeats. Now that's a mouthful, but it does tell you the two main parts found in CRISPR. First of all, we have the repeats. These are going to be short segments of DNA, so 20 to 40 letters in length, and they're going to be palindromes. Remember, a palindrome is a sequence of letters that read the same left to right, like never, odd, or even. So we're going to have these letters that are palindrome. The reason why is that when you transcribe that DNA, you make RNA that forms these little hairpin turns. So we've got the repeats. Those are all identical, one after another, after another, after another, but they're interspaced. And so what's in the middle, we're going to have what's called spacer DNA. Now what's interesting about the spacer DNA is that it's not identical. Each segment of spacer DNA is going to be unique. And this puzzled scientists when they identified this back in the 80s and 90s. But in the 2000s, what they found is that that spacer DNA, that's the important DNA, matches up perfectly with viral, especially bacteriophage DNA. They also identified a number of genes associated with CRISPR. So these are the CRISPR-associated or Cas genes. Now those Cas genes will make Cas proteins.
And we can actually use these Cas proteins to target, for example, only one specific gene. So this method is as efficient as it gets with today's technology. Here is how it works when scientists use these proteins.
These are the two parts of a CRISPR-Cas9 system. This is going to be the CRISPR part. It's going to be the RNA that's got the information of where we want to cut. And then we've got the protein that's actually going to do the cutting. And this is what happens. And so if we've got a little bit of DNA, so this is the DNA that we want to cut, we create a guide RNA that's going to have a corresponding bit of RNA. What happens is the DNA will feed into it like that. Once it's in place, we're going to cut it right here, and we're going to cut it right here. And so we do this little snip, and now we have an inactivated gene. We've broken the gene. Now the cell will try to fix it. It'll do some insertions and deletions, creates mutations. But what we can do a lot of the time is we can inactivate that gene. That's what the bacteria are going to do. But since we've created it, we can cut the DNA wherever we want to cut the DNA. We essentially just have to know what is the sequence of DNA that we want to cut, put that into the guide RNA, and then we can cut it. Now let's say we want to make this more complex. Not only do we want to break a gene, but let's say we want to insert a new gene. Well, now the system's going to just have three parts. We've got the Cas9, we've got the guide RNA, and then we've got the host RNA that we want to put in. So as we break the DNA, the host DNA is going to be added, and then the DNA is going to fix it. So essentially, we've added the gene to the cell. Now, what's cool about the CRISPR-Cas9 system is it does this in living cells, and it can cut the DNA in multiple different places.
Yeah, so now we've seen that genetic engineering with CRISPR is totally safe, and actually, eating GMOs in general poses the same risk as eating the normal food, and we'll get back to the air quotes later. So the people that are anti-GMO are more often than not anti-big ag companies instead of being solely anti-genetically modified food, and believe that the big companies are evil and want to control the whole food supply chain. Now, I'm not saying that's right or wrong, I just want to make the point that when some passionate plant scientists want to make a change for the better in our food supply chain, like these guys at Norfolk, which developed a healthy tomato full of antioxidants using genetic engineering, it's a whole different story than when a Monsanto, for example, is developing GM corn, which can be used with their herbicides and giving the farmers allegedly enslaving contracts for the usage of the corn. So from my point of view, there should be more of a focus on what the scientists are actually doing with the genome, rather than on the fact that one gene out of tens of thousands has been altered. And now to close this up, remember the carrot? Well, the original carrots centuries ago were white, light yellow, or purple. And only after people started messing with the breeding and stuff has the orange one been developed. And since selective breeding is also a form of genetic modification, and basically all of our food was selectively bred for millennia now, it's safe to say that GMOs don't possess any threat whatsoever. Thanks for watching this video.